Thank you again, everyone, for joining us this evening. We have Dr. Daniel Costa with us this evening. Dr. Costa is a distinguished professor in the Ecology and Evolutionary Biology Department at the Institute of Marine Sciences at UC Santa Cruz. His areas of expertise include behavioral ecology, coastal science, animal behavior, ecology, marine biology, ocean studies, scientific method, and natural history. Dr. Costa completed a BA at UCLA and a PhD at UC Santa Cruz, followed by postdoctoral research at the Scripps Institute of Oceanography. His research focuses on the ecology and physiology of marine mammals and seabirds, taking him to every continent and almost every habitat from the Galapagos to Antarctica. He has worked with a broad range of animals, including turtles, penguins, albatross, seals, sea lions, whales, and dolphins, and has published over 400 scientific papers. His current work is aimed at recording the movements and distribution patterns of marine mammals and seabirds in an effort to understand their habitat needs. This work is helping to identify biodiversity hotspots and the factors that create them. He has been developing tools to identify and create viable marine protected areas for the conservation of highly migratory species. He co-founded the Tagging of Pacific Predators program, a multidisciplinary effort to study the movement patterns of 23 species of marine vertebrate predators in the North Pacific Ocean, and is an internationally recognized authority on tracking of marine mammals and birds. Dr. Costa is the recipient of numerous honors and awards, including the Ed Ricketts Award in 2024, which honors scientists who have exhibited exemplary work throughout their career and advanced the status of knowledge in the field of marine science. Today, he brings to us a presentation titled Beyond Pacific Tides: How Technology Has Provided a Window into the Secret Life of Marine Mammals. Please welcome Dr. Daniel Costa.
Well, thank you for the introduction. I assume you can see the slides now? Yeah, okay. Well, thank you for the invitation. Thank you for the opportunity to give this talk. It's kind of an overview, quite honestly, of over my career. I've been I've been fortunate to be part of the development of a lot of the tracking and tracking and technology that allows us to understand what these animals do in the ocean. So that's really what I'm going to try and give you a little bit of insight. I got into this field when I was in high school taking biology. I learned how to scuba dive. And back then, if you look to see what we knew about marine mammals and seabirds in terms of what they did at sea, they used this little funky depth recorder that basically water would go in the tube and wet a dye, and they put those on various animals. And so back then, animals were caught on fishing gear: gray seal, harp seal, northern fur seal, northern elephant seal, 183-meter dive, and then some work on Weddell seals. So this is really where things started, and how we understood animals in the ocean was really rudimentary. And then one of the people that really moved this field along was a guy named Jerry Koyman, who I was very fortunate enough to do a postdoc with. And he took advantage of the fact that up here in the left is this picture of a Weddell seal. These animals naturally are used to going from a hole in the ice and going back and forth to that hole in the ice, because they're living on what's called fast ice, very large expanses of ice, and they'd always return. So here he is as a grad student putting a device on the back of the seal, and the device he used was a very simple clock timer, those little 1-hour kitchen timers that you crank over and it starts ticking away. He put a glass disc on that, which was smoked, and had what's called a Bredone tube, which is this thing here that would scratch on that smoked glass image. And he was able to see that these animals were diving to 600 meters and for making quite long dives. And so this really was the first time depth record was able to get a 1 hour record of an animal diving from an ice hole. Some years later, when I actually was in Jerry's lab, I was able to go down to the Antarctic with him. He had an engineer that built this device that's on the back of a Weddell seal here. And that device looked like this. It used film to record the dive track, and it had a little light emitting diode at the end of a arm, and inside of this tube is a Bourdon tube that would cause this arm to go back and forth as pressure went into the system. And then the film would roll across this. These are little spools here. This is a motor, and that would record a 14-day record. Now, because this was film, we'd have to load this thing in a dark room and hope that we didn't screw up any of the connections and hope that we didn't mess up the O-ring seal. We'd put it on the seal. In this case, it's an Antarctic fur seal, and we also used a harness back then, which proved not to be the best way of attaching it. But that was where we started. And you'd get back this record that looked like this. You can see the light emitting diode tracking against that. The depth first time is going across this image. So looking down this hallway, you can see dive after dive going the length of the hallway. And we would take that film record, expand it, and then we had to sit and digitize this thing by hand. And it would literally take 24 hours of effort to take this on a plotting board and turn all of these data into something that we could analyze. And so it was a pretty laborious prospect. But the other thing is you'd get this record and you'd roll it down the hall and you'd try and think about, well, what was the animal doing? And we would literally make up stories because we look at how the diving pattern changed. I don't know if you can see it here. Their diving pattern's a little different than here. And really it was kind of a fun experience because we had no idea what these animals did. And we were making all these ideas. And really what has happened since that time is that technology has increased. We've really increased our ability to not just tell stories and make speculation, but actually see and get a firsthand view of what these animals are doing. This is that same image in 1983 compared to the same species some almost 20 years later. In this case, the fur seal has a satellite tag that's transmitting to a satellite on board in the horizon. It's telling location. There's a little electronic time depth recorder, and this is a VHF tag, which is the same as what's on here. So you can see over this period of time that things had changed. And the beauty of this is you could sit in your office and get the position of the animal. When you recover this tag, you could download it on computer, and at this time you'd get 3 months of data at a 1-second resolution, which was pretty incredible. And then came along, this is sort of as the technology developed, this was required a titanium housing, and now we had a whopping 512K of memory, and we could use batteries to redeploy this thing. Then the technology continued. The major thing happened here is we're now potting these things in epoxy so that we didn't have to have a machined housing, and that made it a lot cheaper, a lot more reliable. The only downside is it's harder to replace the batteries. But you can see how this technology is both getting smaller and also more reliable. And today there's a whole host of different tags, and this is one of the tags that's used in a lot of the things I'm going to present today. It's a tag that's made at the Sea Mammal Research Unit, glued to the head of a Weddell seal using 5-minute epoxy, just the same epoxy you'd get at the hardware store. So you can see starting with Weddell seals, and in this picture finishing with Weddell seals. What this did is when I first started working on marine mammals, all of our studies were pretty much what was happening on the beach. And this is a female elephant seal with a newborn pup, and about a little bit over a month later, they're weaned looking like this. This is what all of our research was. And all this technology you can see here is a female has a satellite tag on her head and a time depth recorder, that little one I showed you, the Mark 9, on her back. Now we can connect what's happening on the beach to what's happening at sea. And some of the first dive records that we got back look like this. We were able to, this is a 4-day record in the life of an elephant seal. These dark areas are nighttime, the white areas are daytime. And a couple of things you can see is that the animal is diving deepest during the daytime, and then at nighttime it tends to dive shallower. Uh, you can see the dives here are routinely going to 600 meters, 400 to 600 meters. If you remember that first record that people had, thought elephant seals went to 183 meters, that was an unusually shallow dive. We now know that on average, elephant seals can go to 516 meters, which is almost 1,600 feet, with a maximum now for elephant seals of 1,735 meters. That's that's a little over a mile. So can you imagine holding your breath and going for a mile swim? Here's to put that in perspective. Here's that deepest dive of an elephant seal. It was 38 minutes long. Here's the tallest building in Dubai. Here's the World Trade Center. This is 600 meters. This is so the elephant seals are routinely going this distance. If you've ever gotten in the elevator of that World Trade Center, can you imagine going up and down 8 minutes each way, 60 times a day? And each time you do that, holding your breath? It's pretty interesting when you go in that elevator. If you start thinking about an elephant seal doing that routinely, except holding its breath and going to depth and doing a 25-minute dive. So how do elephant seals then compare to other species? One of the beautiful things is we've been able to use these devices on a whole host of animals. And elephant seals really are one of the premier diving animals. They're the deepest diving pinniped, only rivaled by sperm whales and beaked whales. This is all the other seals, sea lions, walruses, other cetaceans. And the only animals that are equivalent to elephant seals are the beaked whales and the sperm whale. These are northern elephant seals, SES, or southern elephant seals. This female northern elephant seal, female southern elephant seals. So you can see elephant seals are comparable to the deepest diving marine mammals, which are the sperm whale and the beaked whales. So one of the other fun things about this is a part of a backstory to this whole thing is the amount of collaboration that has occurred over the course of our studies, working with different colleagues, with different technologies, different devices, different ideas. And one that has been an ongoing one from the beginning, which Bernie LeBoeuf, who started the elephant seal program, was responsible for bringing a fellow named Yasu Naito from the National Institute of Polar Research in Tokyo. And Naito brought all these newer versions of technology that they were developing in Japan. And here you can see under the jaw of this elephant seal is a little device that would tell us when the animal was opening and closing its mouth. And in this figure, this 3D plot, you can see all the little red dots are when the animal was opening and closing its mouth, consistent with foraging, consistent with attacking a fish. And here there's a simple 2D plot of time on this axis, depth here. And you can see these little red dots are predominantly occurring at the bottom of the dives. But then there's a series of dives where there's no jaw movements. You can see them here, you can see them here. And I'll get back to that a little bit later, but we postulated these were potentially processing dives, that the animal was taking a rest from eating and using the oxygen stored inside to digest the food, and or that those were sleeping dives. So we'll come back to that a little bit later in a slightly different perspective. But already now you can see from not having a clue of what these animals doing at sea, how deep and how long, but we're starting to understand not only where they are, because this is a track in 3 dimensions on a map of the ocean, but what they're doing at depth. Now, the other thing we're able to do is figure out, well, what are they eating? And that became an interesting story. We had a variety of different approaches to that. Again, this is where our Japanese colleagues came in with a camera system. And so these are pictures I took off the web. But here's pictures taken from an elephant seal with a little camera on its head, looking at the different kinds of things that it's eating, crustaceans, fish, squids, and other things which probably weren't eating, but that it was seeing. We put these out on 10 seals, 29 hours of video, something like 1,300 different things recorded. And here is a video of an unidentified fish. That's the head of the seal. Boom, there's the fish. And here's another clip. That's a fish that the animal ate. Here's a hake. Yeah. Pacific whiting. Here's one trying to get away. Seal gets it. And that, yeah, so that gives you an idea of I mean, not only are we, I mean, when you think about those videos were taken from an elephant seal in about 300 or 400 meters of water, hundreds of miles, thousands of miles offshore in the North Pacific. Now, the other thing that our colleagues from Japan did, took these data and then started looking at the dive patterns. So all these little dots are actual feeding events, and you can see uh, more feeding events are red in color. And get rid of my phone. These ones down here became really interesting, and these are deep enough that they're into what we call the deep scattering, I mean, the oxygen minimum zone. And so what we postulate is that these animals, when they get down to these deep areas, the prey tends to be deeper. And this is a series of video clips where it looks like the fish that the seal is coming up to isn't really moving very much. And this is something which we know occurs in the oxygen minimum zone, where there isn't enough oxygen to provide oxygen for gill-breathing fish. They tend to go there to avoid being eaten by other fish, but it makes them perfect prey for an elephant seal. So we're actually extending this work because we now have a oxygen sensor that we're putting on the seals to see if we can identify that these feeding events are actually happening when the seals are happening when they're in the oxygen minimum zone. So the other thing that we can do is start to look at how these elephant seal records fit into other studies. And so this is some work that was done at the Monterey Bay Aquarium. And this gets into this whole issue of how habitats are changing. And this is a Humboldt squid, and they have a lot of amazing video from remotely operated vehicles that shows that these squid really start to become more and more apparent starting in 1998 and became very apparent in 2003, 2004, and 2005. And in 2007, I believe it was, we had one elephant seal female that did something that we've never seen happen before or since. We had some 800 animals, and this individual stayed pretty much in the coastal zone. You'll see more tracks in a minute, but spent a lot of time right here. And this area right here is where the fishers were getting a lot of Humboldt squid. This is where the people were finding Humboldt squid. This shows that depth profile from the observations, the Monterey Bay Aquarium, the Mbari people. This is the diving pattern of that female elephant seal, which is actually a male there, but female elephant seal. And we also were able to look at the fatty acid profile in her blubber, which was consistent with eating squid. So we figured that this female elephant seal was taking advantage of this increase of a different kind of prey coming up to the area in the form of Humboldt squid. Now, one of the things that has been kind of fun is to watch how this technology has changed our view of these animals' life history. Back in 1991, this book was published by Marianne Reedman, where she was overviewing what we knew about pinnipeds in general. And this was a map of where we thought elephant seals were. And you can see they're very coastal. That's because this is where we looked for them. This is where we flew our planes, and this is where we drove our boats to make the censuses. So that's where we counted them. I remember one of my colleagues said, you know, Dan, it doesn't make any sense. We count the elephant seals on these islands where the colonies are, and we see many more elephant seals on the islands than we ever see while they're at sea. And he postulated, he says, well, either they're spending a lot of time underwater, or they're leaving the area. It turns out that once we put tags on the animals, both were happening. Here's showing the pattern of tracks for elephant seals. Right up here is from Año Nuevo. Down here is from Isla, the islands off Baja California. So these animals are indeed leaving the area, and they're spending 80 to 90% of their time underwater. This is a little animation that shows the tracks of these animals. They make two trips. One, that's the short one you just saw, which was the post-breeding after they breed. Then they make a longer one. They come back to molt, and then they make a longer trip to sea. And when the little worms, little squiggles are much shorter, that's where the animals aren't moving very fast, and that's where they're feeding. So this shows multiple years of tracking data of our elephant seals. Now, one of the things that we really want to be able to do is use these data for helping us for the management and conservation. And this is a picture I actually took up in off Holland with seeing wind farms offshore, which a lot of people can look at this different ways. Some go, oh, wind farms, I go at it. Oh, cool. We got alternative energy that's not using fossil fuel. Regardless, we need to understand how our animals are going to fit into these habitats. And I think it's important that we have the opportunity to develop these new sources of energy in appropriate ways. So part of that requires us to understand the life history of these animals and when they're going to come into contact with these different new projects or different things that we're doing. So if you take a plane or a ship, you're going to count the animals out there. So on the left, you got humpback whales, on the right, you got some dolphin, and you can see where they are. But what that doesn't tell you is what they're doing. And this is what tagging technology does, is you can see these whales, it's just a cartoon, are moving over a large area, and this larger line is their supposed home species range. So you have whales occupying, individuals occupying most of the species range, which is very different when you have resident species like dolphins, which have a very large species range, but it's composed of many smaller home ranges of a lot of individuals. And why is this important? Well, we need to understand how these animals move, because are they resident where you've got a lot of individuals with small ranges or a lot of individuals with a large range? Are there north and south migration patterns such that you have an area where they're feeding, an area where they're breeding? All of these mean the animals are going to be exposed differently. And in this case, if you just think about on a highly nomadic species, a disturbance of this size will affect all of the animals a little bit, whereas a disturbance on a resident species will affect a few animals a lot. And we're still working out, is one worse than the other? They're obviously going to have very different effects, but it's something that you really need to think about in terms of the natural history of these animals. And our tagging and tracking technology is allowing us to begin to answer these kinds of questions in a way that we never could before. Here's a little example of how we can apply the information we're getting. This is a map I took from the National Marine Fisheries Stock Assessment, and you can see what they plot is the range of the northern elephant seal. And I put a little dot in there and said, okay, we're going to protect elephant seals by, here's an area of the ocean that we want to protect them. But where should you put that area? And a lot of the protected areas are really done by convenience of, well, here's a place we can set aside. It's not based on whether or not it's the best place or other factors. And so we asked the question, given that we know a lot about elephant seals, what happens if we move that marine protected area over the ocean? And so we did that. Here's the range of northern elephant seals. You can see the existing uh, marine protected areas or marine sanctuaries. And here's the range of elephant seals. And so again, you say, okay, here's this whole area, but this is made up of many individual tracts. And so we asked the question, if you protected a very small area and put it anywhere in this region, how many individual elephant seals would you protect? Now, obviously, as you make your marine protected area bigger and bigger, you're going to protect more animals. But again, we wanted to ask the question of, if you just randomly placed it, how good would you be in getting the right amount of animals? How many animals would you protect? And this is what we found. And here's what this says is, with a small area, you're not going to do a lot. That makes a lot of sense. But even when you get an extremely large marine protected area, if you pick the wrong area, which is down here, you could end up protecting very few individuals. You could also get really lucky and protect most of the individuals in that area. What this shows is, on average, if you just say, what's the average outcome, you can reliably maybe get about 20% of the animals. And this just shows you the Monterey Bay Marine Sanctuary is on this size. The I don't try and say that. The Hawaiian Marine Sanctuary is this size, and the Great Barrier Reef is out here. So again, this is just a theoretical conceptual treatment, but it allows us to start to think about how we put together areas that we set aside to protect different species, how important it is that we have the right parameters to say this is an area of the ocean which is important to protect. The other thing that is a collaboration that's kind of great for us is not only can we get our animals to collect information on where they're going in the environment, we can get data on the environment itself. So this is a tag that we put on elephant seals, and a lot of people are doing this on a variety of different species. We put on Antarctic seals, people put on sea turtles. It measures physical properties of the ocean in terms of temperature, salinity, and even chlorophyll. And I said we're now putting oxygen sensors out there. So these data are relevant because the ocean, the way that the ocean works is it's all about temperature and salinity and productivity. So we can look at where the grasslands are in the ocean, where the forest meets the trees, meets the prairies, and that's something that's very hard to do if all you're looking at is the animal movement. So we can get data on the important physical features of the ocean that help us describe the habitat requirements of these seals. The other thing is we have a serious problem with undersampling the ocean. The ocean has weather just like the land. Things move a little bit slower in the ocean, but we've severely undersampled the ocean. We're recently hearing about these hurricanes in the Atlantic. All of those hurricanes are built by temperature patterns in the ocean. And so having our animals collect more information because they're self-propelled and they move through the ocean and they're very inexpensive, we get a lot of information on the ocean. And this is important for understanding ocean weather, ocean climate, and how things are changing. This is an example of how that comes together. That little red thing is an oceanic, basically a hurricane, a cyclone in the ocean. And the seal, this is a track of the seal. You can see the seal really like that gyre, that eddy that forms. This is southeastern Alaska. And we now know that these things concentrate food resources. And you can see here that here's that track of the elephant seal through that eddy. This is warmer water, this is colder water, and this would be a downwelling eddy such that the water is being pushed down into the ocean. So warmer surface water is being pushed down into the deep ocean. And so these elephant seals, and people have done with fur seals and sea lions and other species, allow us to not only see how they're using the ocean, but also understand aspects of the ocean that are otherwise harder for us to get. It wasn't that long ago, gosh, now it's 10 years ago, that we remember the warm blob that occurred off the west coast of the US. Here's Alaska, we're down here somewhere. These are tracks of elephant seals that we had before and during and after the occurrence of the blob. And you can see that the elephant seals were going right through this area of the warming ocean. And what was really valuable about these elephant seals is we knew the blob existed because mostly of satellite imagery. So we knew what the surface characteristics of the blob were, but there weren't a lot of oceanographic measurements. There weren't ships going out there. They had a few buoys, but the actual depth and structure of this feature wasn't really well described. Well, since our elephant seals were going through that feature, we were able to define, you can see this is a track, that track of that elephant seal I showed you moving through this region, and you can see that warm surface layer of the blob going actually fairly deep. You can see that that's a fairly deep feature in the ocean. So we were able to use these data to then to actually look at a much more robust image of the characteristics of the blob and understand it in a way that we couldn't by just using satellites. Now, the other thing that's been quite fortunate is I started, with Bernie LeBuff and myself, started working on elephant seals many years ago, where we we're measuring weaning mass of animals. And so you can see measurements of weaning mass. It goes back to the 1980s. And we've been continuing to measure these massive weanling elephant seals up until present. We look at the pup production. And then these are the characteristics of the Pacific Decadal Oscillation and the El Nino Southern Oscillation. These are characteristics of the ocean in terms of warming and wind patterns. But what's important to note is that the number of pups born has increased, but the weaning mass of the pups has decreased. And then there's a fair bit of variability. And we now know that this variability is tightly connected to what's happening in the ocean. It's a combination of a lot more elephant seals, but also these changing ocean conditions. And part of that is that we've been tagging as part of the tracking of Pacific predators program. We started a big effort in 2004, putting out 20 tags every cycle through present. So we have the data on what these animals were doing. So this is the horizontal distance traveled, and this is the total distance on the order of 3,000 kilometers. So since 2004 to 2020, elephant seals are working harder. They're going farther out to sea to find resources. And so we're not entirely sure what this means yet, but it really does show that there is this long-term change in how the elephant seals are using the ocean and are responding to the changes in the ocean environment. Now, one of the things that I had a graduate student, Samantha Simmons, who was really interested in, do elephant seals go back to the same place? And we had some ideas that maybe 1 year, if things were really good, they'd go back to the same place. These are tracks of individual females for, in each case, the same post-molt season. So this you can see, we did these on 45 different females, same season, repeats. So this is one female that over 3 different years, she was doing pretty much the same thing. Here's a female that over 2 years was doing something very similar. And this female over 3 years, 1 year she went way far out, 1 year up the coast, and 1 year further out. So we've been looking at how elephant seals are using the environment. And then there is this example, which was interestingly enough an accident. We put a tag out on this female in 1995 and got this track. One of my students was looking for 6-year-old females. He read the tag wrong, put a tag on this female in 2006 who is now 17 years old. But by accident, we tagged the same female we had put a tag on 11 years later. And what's amazing about this is that here's a female as a 6-year-old in 1995, that same female 11 years later as a 17-year-old that seems to be doing the same thing. Now, what we think is happening here is because we've done a larger sample comparing, not all the females do this. Some females have high site fidelity, others don't. We're still trying to figure out what drives this. Is this something that is learned? Because we have females that don't do this at all. And the females that do this are the ones that tend to forage in areas that are very reliable and very predictable. So on average, this is a good strategy. There are other females that are much more variable, and those females do better in a much more variable ocean. And so that's an ongoing piece of work that we're continuing to explore. Now, one of the things that, I really enjoy working with bright, young, creative people. And one of my graduate students, Sarah Peterson, said, I want to study mercury. And we had some people study mercury levels. And I said, yeah, okay, go ahead. I wasn't sure it was going to be very interesting, but she went and looked at mercury levels, and she worked with some people that are experts in mercury on elephant seals. And we're able to take samples from the pup. We also did some work with females, but we took samples from the pup, which reflects what's in the female. And this is the mercury levels in elephant seals. This is mercury levels in all kinds of other marine mammals. And this harbor seals and stellar sea lions have high mercury levels. But elephant seals have really high mercury levels on average, and with a few individuals having extremely high levels. Now, a lot of this has to do with the fact that elephant seals feed on those deep mesopelagic fish. And deep things in the mesopelagic zone are naturally high in mercury. So if you eat tuna or swordfish, remember those are animals that people worry about mercury levels. That's because those animals are feeding in the deep ocean, which the marine chemistry of pressure and the characteristics of that situation creates an environment which mercury is much more bioavailable. So from one perspective, we shouldn't be surprised that elephant seals have high mercury levels. Fortunately, we don't eat elephant seals. But we took this a step further and then asked, well, do the different places that females forage, do we see different levels of mercury? And in fact, we do. So this is the levels of pups of females who foraged in the Gulf of Alaska. This is females that were foraging in the sort of in the in this part of the North Pacific, and we call this the transition zone. And these are females that were foraging deeper. So the yellow is shallower feeding females. This is deeper females. So the deeper females are feeding in the area where the fish are going to be most uh, are going to be have access to the greatest amount of mercury levels in the ocean. So part of this is because it's naturally high in mercury. But the other reason that this area is high in mercury is because the winds move from China across the North Pacific. And a lot of the coal-fired plants and a lot of the industrial activities that put mercury into the air are going to come across with these winds and get layered into the ocean and then absorbed into that habitat. So there's a combination of naturally elevated mercury levels, but also human introduced mercury to the North Pacific. So we can see that now elephant seals are also telling us something about the pollutant levels in the North Pacific. Okay, I've spent most of my time talking about elephant seals because we've done a lot of work on them. I'm going to switch to a study that the first 10 years of the 2000s, from 2000 to 2010, with Barbara Block with funding for the Census of Marine Life, we had a program called Tagging Pacific Predators. And this was a little silly figure I produced because this really was kind of what we knew about the movement of a variety of marine vertebrates. We had we hadn't done a lot of tracking. We kind of suspected this is the movement patterns. And so top was all about tagging animals. These are salmon sharks, these are bluefin tuna, elephant seals, and albatross. We wanted to see what their movement patterns were. When we finished that study, we published this paper in 2011, from that silly little cartoon to a much deeper view of the animals that use the North Pacific. And these down here are sooty shearwaters. These are leatherback sea turtles. Leatherback sea turtles here. There's tuna going back and forth. There's salmon sharks. Anyway, we got a view of multi-species use of the North Pacific, which was a first and quite exciting thing. We also found out that the areas where these animals spent most of their time was in the North Pacific transition zone. You saw I was talking about that with the elephant seals, the California current. We kind of expected this because this is where the most upwelling is. It's sort of the grassland, the Serengeti we call this the blue Serengeti. We knew there's a lot of productivity here. This shows the California current where the density of all of those top critters is greatest. What we didn't expect was the White Shark Cafe. And this was an area nobody knew about. We only found by putting tags on white sharks that this is where white sharks are off our coast here in about now, coming in fall. And then in the beginning of winter, they disappeared somewhere. A lot of them go over here, but they spend a lot of time in this area. I'm going to pivot now to working with two of our pinniped species. I'm going to talk about sea lions and elephant seals. I'm going to talk about them in the context of being prey of these two predators, of killer whales and white sharks. And this is a new project that with Barbara Block, we have the Monterey Bay white sharks, where we're looking at the relationship of white shark movements relative to their prey, which is elephant seals, sea lions, and to a lesser degree seals. And so here's our sea lions on top of elephant seals. That's my segue into focusing on the differences between these two Very interesting animals and how they potentially deal with predation. So you can see here's our team putting a satellite tracking device on a sea lion and on an elephant seal. A lot of the same technology, just slightly different tags or different ways of catching the animals. This just shows you these are really different animals. And to me, this is one of the exciting things about this work is these white lines are what female northern elephant seals do. These red lines are what female California sea lion females do. So the areas of which they move is very different. This shows you the depth and the dive duration. And you can see that elephant seals, as we've already seen, are champion divers. These are all a bunch of dives of females, and the red are California sea lions, females. So you see that these animals are using the environment in incredibly different ways, and it allows them to avoid predation or not in different ways. So one of the things that elephant seals do, and most of the white sharks, we know most of the predation occurs near the colony. What better place to be to find your prey where they're concentrated, which is right off the colony. And so we know that that's where the white sharks are because we've tracked them. When an elephant seal leaves the beach, here it is sitting at Año Nuevo. It pretty much follows the contour of the ocean. This is the continental shelf, and then it goes into the deep ocean, and it's diving much. The ocean is much, much deeper here. So we think that this is probably a predator escape mechanism. Here you can see the departure and return of an elephant seal. So this is a very small proportion of its trip, and all this time is out in the pelagic ocean. So the other thing to note here is that when does the animal sleep or take a rest? There's almost no period of time that it's spent on the surface. These are 20, 25 minutes underwater, 2 to 3 minutes on the surface, 25 minutes underwater, 2 to 3 minutes on the surface. 80 to 90% of the time is spent underwater. So the time that they're exposed to white shark predation is very small, but it's when they're leaving the island. We also know from colleagues Josh McInnes that a lot of killer whales take elephant seals in Monterey Bay. We don't really know what's happening out here. We don't know if killer whales are taking them out here. But again, what better place to find prey that when they're but other than when they're concentrated. So one of the things that we think about in terms of why do elephant seals, how do they avoid predation when they're at sea? We know there's all these foraging dives, and because we had that little recorder on the cheek, we think that these are processing dives and or sleeping dives because if you want to stay away from the surface, how do you then spend the time digesting your prey? Many of us, I mean, think about when you have a big nap, when you have a big meal, you go and you sleep and you just relax. Well, we think that elephant seals are processing their food using the oxygen that would have been used to forage to support their muscle movement they sink down to depth and then just sink. And we think that all that oxygen that would have been used for swimming is now being used for digesting the food. We look at that because here's a typical dive pattern. When a seal is skinny, it sinks. And as the trip goes on and they've been feeding, they get fat and now they tend to go to depth and float up. So their buoyancy changes because they become fatter. And I kept, for the longest time, that is where the story would end. That we'd say, well, we're not entirely sure what these trips are about. We think they're processing, they could be sleeping. But I had a graduate student, Terry Williams, and I had a graduate student named Jessica Kendall Barr, who had been involved in some sleep studies before as an undergraduate working in a lab. And she wanted to come and study sleep in elephant seals. And we said, okay. We got a device that had been a person in Switzerland had been developing a neurologger that could measure EEGs and ECG of all those parameters on animals remotely, a little tiny device. Jess built a cap that we could just do surface mounted electrodes because what those guys did would drill a hole and put those electrodes in the brain. We weren't going to do that. We were able to put this thing in an underwater housing and basically adapt it to work on elephant seals. And so she did that. We had an elephant seal going down at depth. 23-minute dive. You can see it's one of these drift dives. The animal is actively awake, goes into that drift phase of the dive. It's quiet waking. It starts slow wave sleep, deep slow wave sleep, and then it starts to go through these little floating corkscrews, and that's apparently when rapid eye movement sleep occurs, followed by deep slow wave sleep, coming up and then awake. And so it looks like elephant seals are indeed sleeping during these drift dives. And that's the only time that we saw them sleeping. And here's an example of an elephant seal. That track I showed you here is diving, diving, diving. The animal's diving. And finally, those drift dives, we now know that those are dives that are also sleeping. And when we calculate how many of those windows of sleep are occurring, elephant seals only averaged about 2 hours of sleep per day for 7 months. The only other mammal that's anything like that is an elephant. So maybe elephant seals and elephants have something more in common than just their name. Now, one of the other interesting things about this is if you think about elephant seals are really good at avoiding the surface where predators might be. And one of the things that we found is every now and then, and on some females more than others, we'll get a series of dives that are extremely long. So here is a record of a female that has a 2-minute surface interval, an hour and 9-minute dive, 2-minute surface interval, hour dive, 51-minute dive, an hour and 43-minute dive. What is amazing is all of these surface intervals of just 2 minutes. This is that longest duration dive, and an hour and 43 minutes. Can you imagine holding your breath for an hour and 43 minutes? And then what doesn't fit any of our understanding of physiology of these animals is that these dives, that these animals are able to come up to the surface and completely replenish their oxygen and or deal with other metabolic end products, which that's the part we don't understand. They should have a lot of lactic acid built up, but apparently they're doing something different. And this is something that we don't really understand. But in 11 hours, that female spent only 20 minutes at the surface over 11 dives. Normally, she would have made 3 20-minute dives an hour or 66 minutes on the surface. So her exposure to what is ever happening at the surface, whether it's a predator or some other disturbance, she's reduced her exposure by what to be 1/3 of what it would otherwise be. So this is an ongoing thing that we're trying to tease apart and get a better understanding for. What do sea lions do? Well, sea lions don't have any of these tricks. They're much faster and more agile than an elephant seal. Elephant seals are kind of quiet. They swim alone, swim along the bottom. When sea lions come to and from the island, they swim pretty fast. And we've been putting tags out on sea lions at Año Nuevo. These shows tracks of females with pups. These are juveniles. Now, realize Año Nuevo is a little bit south of San Francisco. So we're up here, just north of Santa Cruz. We had 2 little subadult sea lions that went all the way down to Baja California and back. So that is kind of, I mean, for a sea lion, for an elephant seal, that's nothing. But for a sea lion, that's pretty remarkable. We also had one female that went down to San Miguel Island. And our speculation is that she probably has a pup here because she actually made multiple trips here. She probably came to Año Nuevo to rest from her pup and then went back said, okay, I'm not coming back there. And she went back to to San Miguel where we think her pup was. We're using these tracks, we're able to think about, well, what do sea lions do when they come to and from? There are a gauntlet of white sharks off the island. This is underneath all these green lines is Año Nuevo Island where the California sea lion colony is. The white sharks are right off here. And so if you look at what a sea lion does on the island, they're not going anywhere. When they depart, they swim rapidly. And then when they get to where they want to feed or moving, they're kind of moving around normal speed. When they return, they swim rapidly to get onto the beach. When they leave, they swim rapidly offshore. Then they're at sea, come back. You can see that it appears that, not surprisingly, the way they do this, they swim at the surface very rapidly, possibly outswimming white sharks. These are some tracks of one of our females that had a pup here at Año Nuevo that was going up to see you guys. Another one that was sort of moving offshore. This is an ongoing project. This is that female that probably had a pup on San Miguel that this is a pretty remarkable trip, for multiple trips for a female with a pup, California sea lion female. And again, these are new data that we're still working out. Through in this slide, George Colasso is a graduate student with me, is working on these tracking data. We're also using drones, and I thought I'd throw this in because drones are the big thing people talk about. This is part of this study is to quantify the seasonality of sea lions along the colonies and the haul out areas within the white shark's range. But one of the things you got to be careful about is sea lions change their behavior as a function of daytime. So this is 8 a.m. All these are machine learning identifications of sea lions. You can see by 10 a.m. the area up here is starting to decline. And by noon, most of the animals that were hauled out up here have moved down to the cooler peat parts of the beach. So not surprisingly, sea lions are moving. When it gets warm, they move closer to the cool water. And in the nighttime, when they're cooler, they move up onto the shore, up onto the larger parts of the island. So this is, again, just a little snippet. There's so many other things that drones are really opening up different avenues, and I'm not even going to touch on that because it's really too much to talk about. As part of the tagging of Pacific predators program, we tagged a bunch of different species, and again, that's a talk in and of itself. But I'll just mention one. Many of you see these shearwaters swimming off the coast and flying off the coast in these incredibly large flocks. These guys breed in the islands off New Zealand. And this was some work that Scott Schaefer at San Jose State was the lead on. These blue tracks are during the breeding season, where the shearwaters have chicks on nests on the island south of New Zealand. We track these birds, and so at the end of the austral summer, the southern hemisphere summer, they sort of fly to the west and then come across and spend their summer up here off the coast, what we see up off Alaska and off Japan, and then turn around and fly back to New Zealand. And so when we published this paper, we called it The Endless Summer. This shows you the tracks. The animals are in New Zealand feeding, supporting their chicks. They now migrate across the Pacific. And if you were a sailor, this is the track you would take, because they're using the wind to help them make their way across the ocean. And they're doing their feeding in our summer, and they get topped up and then return very rapidly to begin the breeding season in the islands off New Zealand. Okay, so again, using some of the data that we have collected under TOP, which was tagging a variety of different species, we decided to ask the question, how do our data help us understand the efficacy, the effectiveness of sanctuaries? And so it would not surprising, elephant seals are moving so far off in the ocean, they'd be hard to protect. But the good news is that a lot of the breeding colonies, and almost all the breeding colonies in elephant seals, are in one sanctuary or a marine park. So while their offshore region isn't really protected, their breeding colonies are. This is tracks of blue whales, of work that Bruce Mate did as part of TOP. And you can see that the Monterey Bay National Marine Sanctuary does cover a fair amount of the habitat that blue whales are foraging in and during their summer-fall trips. With humpback whales, a lot of their foraging habitat, we have much more data that we haven't updated this figure in a while, but it does a good job with humpback whales. We looked with California sea lions, and we didn't have as much data up here, but again, we can see a lot of their breeding colonies are covered by the sanctuaries and national parks, national monuments. I'm now going to switch because we started talking about conservation, and I just wanted to move to show how these tagging and tracking technologies have been used in a variety of other regions to address important questions of conservation. This is one of my favorite sea lions. This is Australian sea lion female and her pup. You can see we put tracking devices on mom and on the pup. We were interested in seeing how the development of the pup goes, and does the pup follow mom. And uh, we had earlier found that this animal has a very the foraging foraging patterns of this animal put it in an extreme environment that it's really working very, very hard. And we found that the pups stay with mom longer than most other sea lions because it takes a long time to develop this capability of carrying out the life history of an adult Australian sea lion. But along the way, we recognized that our data could be used for conservation and management. And Simon Goldsworthy, who was working with the South Australian government, uh, was able to identify that there was a shark fishery off the coast of where these animals are. And these are images of female Australian sea lions that were caught and killed in the shark gillnet fishery. Simon and colleagues took a lot of the data that we collected, and the work I did was right here off Kangaroo Island. They put tags out on the other colonies, and we had the tracking data of the sea lions. This is the work that our lab did. We looked at the range of the gillnet fishery, and you could see that there is a significant overlap between the gillnet fishery and the colony where these animals were breeding. The other important thing is we knew that these animals were feeding on the continental shelf, and that allowed us to create a very clear area where if you prevented fishing on the continental shelf below if you prevented fishing below 100 meters, you would pretty much save Australian sea lion habitat and avoid any conflict. There's a long process, but the end of the story is Simon Goldsworthy and colleagues and the Australian Fisheries Management Agency essentially banned the gillnet fishery from the regions off the colonies. And I think this was an animal that there's about 5,000 females scattered across the southern coast of Australia. So it really was a significant conservation management milestone. They were losing 50 females a year, and a population of 5,000 that has a very low reproductive rate can't sustain that. I'm now going to take you to the Antarctic, where there's some other work that, again, collaboration has been the key to this. And we did a lot of work in the Antarctic Peninsula region. The Antarctic is an amazing place to work. We were looking, again, this idea of looking at multiple species. We've tracked Antarctic fur seals, Weddell seals, leopard seals, elephant seals, and crab-eater seals, all using these satellite tracking technologies. I'll just say, of all these animals, the last one I worked on was leopard seals, because probably obvious. They're named leopard seals for not just because they have leopard spots, because they're an apex predator. They eat everything. So we worked out working on these other. Weddell seals are really easy to work with. Elephant seals we had lots of experience with. Crab-eater seals were interesting. They're a little feistier, and fur seals are quite feisty. But we tagged these animals with support from the National Science Foundation and from the U.S. Antarctic Marine Living Resources Program. That's working on a leopard seal. That's working on a southern elephant seal. This underneath all those people is a crab-eater seal. This shows you leopard seal. This was the smallest one. This was the larger one. We found that females are much larger than males, and that's not unusual for some of the faucid seals. Weddell seals are the same, but females are significantly larger, and that's an interesting thing that we're still sorting out. But the end of the story is that these animals are using the habitat in very different ways. You can see the red here, the crab-eater seals. The green are elephant seals. This mustard is fur seals. You can barely see the Weddell seals in there. And this particular figure didn't have our leopard seal data in it yet. Well, it did, but you can't really see the tracks of leopard seals. If you look at I showed you this picture for sea lions and elephant seals in the Pacific. These animals are using the habitat in very different ways. You can see horizontally, spatially, they're using different places, and vertically. So this is a leopard seal, which is feeding relatively shallow. They feed on krill, on other seals, on penguins, on fur seal pups. Crab eater seals are a little deeper and a little longer. Elephant seals are the deepest, longest, and this is the mean depth and maximum depth. And so you can see these animals are both partitioning the habitat horizontally and vertically. I could tell you that these animals feed on different things, and so that all makes sense in terms of they feed on different things and they go to different places. Part of what I find interesting is, okay, these animals have these different feeding behaviors and different diving behaviors. What's the physiology behind that that allows them to do that? So in this figure, I show you the mean dive duration. So this is how good a diver they are. And on this axis, it's how big is their scuba tank? What's the oxygen stores that they have on board? And this is stored in their blood and their muscle is sort of, I like to think of as their internal scuba tank. And so you wouldn't be surprised, but what I think is cool about this is it shows that the physiology and the behavior are tightly coupled. So for these animals that aren't diving very long and very deep, the leopard seal and crab eater seal, they have reasonable oxygen stores, but not great. For elephant seals and Weddell seals that are diving long and deep, they have much larger oxygen stores. So the elephant seal, again, was the deepest, longest diver. It has the largest oxygen stores. And Weddell seals aren't bad. They have a pretty good internal scuba tank. But these two guys are the really excellent. You can see again that those are the deep, long divers, and these other guys are the shorter divers. So the physiology complements the behavior or enables the behavior, if you will. Taking this a step further and asking, well, we know that the Southern Ocean is changing fairly rapidly, and we know that the sea ice extent is starting to decline. And the problem with that is the sea ice is the home and the habitat for krill, and krill is a major component of the food web of the Antarctic. This is a picture of the teeth of a crab eater seal, which has these crazy teeth. These teeth help it act as a sieve, and it goes and feeds just like blue whales and humpback whales. It sieves through the water, feeding on krill. And so this is a krill specialist, and we can tie in the extent of sea ice with krill availability, and that makes us concerned about crab eater seals. Is their habitat going to change? These are red dots are individual elephant seals. These green dots are individual crab eater seals. And the point of this picture is that you see the red dots are moving all over. They're moving in the pack ice. Now they're going to South Georgia and the Falklands to breed. But the crab eater seals are staying in the ice. So the crab eater seals are staying in that ice habitat, and elephant seals are moving all over the place. And I take this to suggest that elephant seals are going to have a much easier time responding to climate change, to the reduction in sea ice, and that crab eater seals are going to have a harder time, and their population is most likely going to decline. I mentioned to you that this collaboration where we work with our colleagues and the physical scientists, physical oceanographers, and this is another example of that. We've been putting tags out on elephant seals up here on an island in the Antarctic Peninsula. And in 2008, in this region here, is when the Wilkins ice shelf collapsed. This is in February, in March, and in May. You can see this whole ice shelf. This fell off. It was a nice solid ice shelf, and it sort of came off and made a lot of icebergs. I knew I had all these data of seals that hang temperature profiles, and so I put out the word that, is there anybody out there that could use these data? And a guy named Laurie Padman said, sure, give me those data. And so he started pulling the data together. And he also found an interesting thing was that he said, well, these elephant seals are diving deeper than the bottom. And I said, yeah, no, they're not mole seals. We know that the bathymetry of this region is not very good. The ocean bottom is not well mapped. It's not surprising. It's a hard place to get to, and it's based purely on satellite altimetry, which averages the ocean depths, and it's a very sort of magical thing that they do. He was able to map the ocean bottom using our elephant seals. Whenever the elephant seal dove deeper than the bottom, we knew that the bottom was deeper there. So we modified the maps. And what that did is it showed us that there was a canyon here and a canyon here, and that canyon was allowing warmer oceanic water to come up, whoops, to come up underneath the ice shelf. And you can see this is warm water coming up here, and you can see the colder water. This is the temperature. The blue is colder water. So that warm water was coming up underneath the ice shelf, warming it up, cooling that water, and that helped cause this ice shelf to collapse sooner than the atmospheric models would have anticipated. So this became what's known as basal melt, and it's an important component now of understanding the ice shelves are much less stable than we originally thought because of the increased heat coming from the ocean, and it's not just the atmospheric heat. Okay, we're on the home run here. Collaboration again is important, and a bunch of us as part of the Scientific Committee of Antarctic Research expert group on marine mammals and seabirds said, let's get together and put all of our tracking data together. And we created this program called Retrospective Analysis of Antarctic Tracking Data. And we got all these folks to contribute their data, and it was a core team of us that pulled all these data together. And we wanted to see a lot of the things I've talked about. This was to look at that as a multi-species, full Southern Ocean view. And here's an animation showing all of the data that we got for all of these different species using the Southern Ocean environment. And so it became a really exciting opportunity. It took us quite a few years to get the data, to harmonize it, to get it, because everybody collects their data in slightly different ways. And so big major task was to get the data and then get the metadata and then put it all together. Once we did that, we had an amazing data set, and we had some Ryan Reisinger, who is an incredible individual, did a lot of the analysis. And what we're able to do is find the hotspots. Where are the biodiversity hotspots of the Southern Ocean? You can see here and around the Antarctic continent. Interestingly, this is sort of a cold spot. The South Pacific is an area where we don't see a lot of animals. A lot of that has to do with more islands out in here and a lot of the aspects of the bathymetry. But we then defined these these hotspots, these biodiversity hotspots, and then asked the question, how much of those areas are protected? And the interesting comment here is a lot in the Southern Ocean, a lot of the protected areas were based on tracking data. So we had really good defined marine protected areas. And they then said, all right, here's the current marine protected areas. And this is the areas that exist today. But then we asked the question, as the climate changes, how will these marine protected areas, will they still do as good a job as they're doing today? And this figure here says that overall, we're going to lose habitat, that the habitat is moving outside of the marine protected areas. The greatest area that we're going to lose protected habitat is in the Antarctic, because it's not surprising that the habitat's constricting. The areas that are going to increase are the subantarctic. So these subantarctic islands are actually the habitat's going to move more into the areas that are protected. And then we can also ask the question about current and proposed, but the idea is taking these tracking data and understanding where are the important places of the Southern Ocean. And why are we working in the Southern Ocean? It's one of the areas that is internationally, a lot of international collaboration. There's the Antarctic Treaty. There's the Convention on Conservation of Antarctic Marine Living Resources. So it's an area where there is already the willingness to address these larger questions of conservation and management. And so it's a great place to do this kind of work because we can make a difference in there's prove a path forward so that we can apply these to other regions. And that is a bit of a taking you from the beginning of where we were studying animals with very primitive techniques to uh, to what we're doing now. And really, I just touched the surface on the technologies and all of the different tools that those people who study uh, marine vertebrates are able to use. So I'm ready for any questions.
Wow. Well, thank you, Dr. Costa. That was fascinating work that you're doing, really important work that you're doing. So I know that there's so much more you could have shared with us, but we really appreciate um, hearing your work. There are a couple of questions that came up in the chat. I'll just read those quickly, and then if people have questions, just raise your hand, and we can get to those as well. We had somebody ask, any idea what happens with the proboscis when an adult male elephant seal is fishing? Does it go up and out of the way or to the side?
It's a
Curious.
great question. What I can tell you is that during the breeding season, the nose, the proboscis actually gets a little larger. And so we don't really know what's happening, whether it's engorged with blood, so that the really big nose that you see when the animals are on the beach does get a little bit smaller once they're at sea, but it doesn't go away. I mean, it just gets a little bit smaller. We don't really know how that works, but I can tell you that we just deployed some cameras. So those cameras that I showed you the video uh, were all done on females. Our Japanese colleagues came with some cameras and we're now putting them on the males. And so maybe we'll be able to answer that question in the near future. But great question. Honestly, I don't know.
Oh, very good. We have another question. Are there any theories about how and when the northern and southern elephant seals parted ways?
We've been that's yeah, it's interesting. There isn't a good fossil record, but we do know that we know that elephant seals as a group, we see them in the a couple of million years ago. There's some fossils off South America. I don't believe we're not really sure whether a lot of people think that elephant seals were derived in the Southern Ocean and then probably during the when the Isthmus of Panama closed, it sort of made the oceanic patterns a little maybe more conducive to things moving into the north. A few individuals probably came across. We do see southern elephant seals. There's at least 3 or 4 sightings. There was a recent one in the Sea of Cortez, the Gulf of California, of a southern elephant seal that came up here. There's been some others off various parts of Mexico. So we know that even modern southern elephant seals have come into the North Pacific. So they're very, very similar. They're, having worked on both species, they, their behaviors are quite similar. They're genetically very similar. The southern elephant seal never went through the bottleneck that the northerns did. The southern elephant seals were hunted for their blubber, for the oil, but there were always more of them than northerns, and they were also distributed throughout more of the Southern Ocean. So southern elephant seals went through several bottlenecks. Northern elephant seals were reduced to less than 30 individuals on Guadalupe Island by 1900. Uh, so yeah, we best guess is probably 1 or 2 million years ago. Uh, group of southern elephant seals immigrated into the north and gave us northern elephant seals. Most of the elephant seals are in a group called Monachian or southern phocids. So they're closely related to the Antarctic seals, to the Southern Ocean seals. So they probably did evolve in the Southern Ocean in the Southern Hemisphere and then come up to the north. But that's about as much as we We know I have a former student who's a paleontologist, and I just recently asked her that question. And so that's what I told you is what she told me, pretty much.
Well, very good. We have a fishing question here. Is there any concern that fishing fleets may target new areas using the new data? Has this been done in the past? And how long do the devices stay attached? And how are they retrieved? Any comments on the krill harvest this year and the future of krill fishing and protection? Those were a lot of questions in
A lot of
there.
questions. Okay, so in terms of we're not worried that people are going to use our data to figure out where to fish. Uh, we are using our data to maybe get a baseline. Roxanne Beltran was the lead author on a science paper, which did just that. We estimated in that paper how much food the elephant seals are feeding. We were able to show that it varies from year to year based on these oceanographic patterns. Nobody's currently harvesting these deep mesopelagic fish. People have done studies using echo sounders and shown that there's probably 10 times or more biomass down there than we ever anticipated. So there is certainly the possibility that this might be used uh, as a fish resource. It's deep. It's not the easiest place to go and fish. And you saw those images. They're not particularly attractive fish for people to eat, so they would probably be harvested for fish meal. But that is a concern. So it's not that they're going to use elephant seals or our tracking data to find the resource, but we're now starting to at least get a baseline of what these animals are doing beforehand.
Okay,
The,
kind of related, any comments on the krill harvest this year and the future of krill fishing and protection? Is that
that's where, yeah, the krill harvest is there's a couple of things happening there, and one of them is we have climate change that is changing is making the habitat less beneficial to krill. You also have a fishery which is going after krill, and the area that I showed you, the Antarctic Peninsula, is the area where most of the krill harvesting takes place. One of the other aspects of that is that the Norwegians have come up with a relatively new method of krill harvesting. Traditionally, what they would do is use a trawl net, and they would run it through the water and then pull it up and empty it, put it back in the water, and run it for 20 minutes and then pull it up. The Norwegians have figured out to use those trawl nets, but to basically connect them to the surface with a tube that basically vacuums the krill that's caught. So they don't have to bring those things up. So basically, they've got an incredibly more efficient way of harvesting krill. You put that on top of the fact that the climate is getting less favorable for krill. We have a number of people here at UC Santa Cruz that have been studying. Ari Friedlander and his group have been studying humpback whales now for several years, well, quite a few years. Uh, we One of our students is now finished, looked at aspects of stress hormones and reproductive hormones, and we are seeing some changes in the reproductive parameters of humpback whales. I have a student that did a modeling exercise where she looked at the energetics of humpback whales and modeled both the krill fishery and the climate change projections. And I guess the sad news is the climate change is by far the most worrisome component of the krill fishery. That's not to say that the krill fishery isn't a problem. It's just that what will happen in terms of the future climate change is going to have a much stronger impact than the krill fishery. What the problem for the krill fishery is going to be is that, okay, now you've got climate change causing a reduction, causing problems for these critters, as well as the krill fishery. So it's going to be very challenging for CAMLAR, the organization, the international organization that manages krill fishing. And one of their fundamental concepts is to make sure that the krill fishery does not impact the mammals and birds of the Southern Ocean. And but it's a consensus body. That means everybody has to agree on whatever management decisions. They've put in a marine protected area where there's a no fishing zone in the Ross Sea. There's proposals to do that for the Antarctic Peninsula. It took them 10 years to get the Ross Sea one. And that is an area that was mostly about the toothfish fishery. So yeah, that's what, it's a problem and it's one that needs resolution.
Okay, oh, very good. Well, we have a few more questions that are coming in here, and I know we're kind of running a little low on time here. I just want to make sure we get to everybody. So there was still another question from this person. How long do the devices
Oh yeah.
stay attached, and how are they retrieved?
So they don't stay any longer. You can see in the picture that it's sitting on its head, and you can see that little stuff at the base. That's 5-minute epoxy. So it won't stay on any longer than, depending upon when we put the tag out. If we put the tag out immediately after the animals have molted and grown back new hair, the tag will last a year because it'll fall off when the animal molts, when it sheds its hair. Tags usually don't last that long. They usually, with elephant seals, they do. Elephant seals are really excellent. For the Waddell seal, the picture we have here, uh, that tag transmits the data via satellite. I guess it didn't really say that. The information is transmitted on diving behavior, temperature profiles, all to an Argo satellite system. And we don't have to recover the tag. If we are lucky enough to recover the tag, we have much more data because the satellite system is very limited in its in how much information you can transmit. One of the reasons we have such a rich data set on northern elephant seals is because 80% of our females come back to the beach where we tag them, 80 to 90%. So we put tags on females, they go to sea, they come back, and we recover the tag, put in a new battery, pull all the data off. So that's why we have over 1,000 tracks of female elephant seals, and it's only because we have this colony 30 minutes north, 15 miles north of campus. We can go up there, put tags out, animals come back and recover them. That's been why we've been able to do so much with elephant seals. For the sea lions, we put the tags on them, not expecting to see them again, but occasionally we get lucky, and we've had 2 tags fall off on Año Nuevo Island, and we have 2 tags that are sitting on the Farallon Islands. We couldn't go and get them during the bird breeding season, and we're hoping to recover them in a few weeks. And we get much more information when we get the tag recovered, but that's how that works.
Very good. Thank you. We have a question here about asking if there's anyone who's thinking about tracking right whales. They're occasionally spotted in the northeastern Pacific, but their habitat isn't known. Would it be possible to attach satellite tags to them when they do show up to see where they go?
Well, the answer is it's definitely possible, but the counter answer to that is that there's a lot of reluctance about using the current tagging technology on right whales. They have Bruce Mate from Oregon State University has tagged a few right whales, and the way that we tag large cetaceans is they don't have hair, so you can't glue it to their hair, and it's some combination of either a little uh, dart like little projections that you basically it sticks into it's you stick it on into the animal, and some of those tags will last a few weeks. The ones that last much longer are ones that actually you shoot into the animal, and it they've done a lot of that with blue whales and humpback whales. The tagging technology is quite excellent. There's with when you're down to a few hundred northern right whales, I don't know where we're at now, somewhere probably around 300 plus or minus. There's a real reluctance to use those kinds of uh, tracking technologies. And there's different perspectives on it, and we people have been using those tags on southern right whales, and so we have tracked southern right whales. But yeah, I mean, it's the technology is available, but there's reluctance to use it on such a highly endangered species is the answer.
Understood. Very good. So Sarah Grimes asks about how many California sea lions are born on Año Nuevo each year.
That's a great question. In 20 years ago, two or three was a big deal, and that's what we today. There's over 700, so it's we have over 700 pups born every year at Año Nuevo, and the same for the Farallons. And this is something that's happened over the last 20 years. Is that uh, the sea lions are pupping more up on these Central California islands. We're not sure whether the sea lion population also has been expanding. So in 2018, the expectations were that California sea lions had reached their carrying capacity. And in the 1970s, the population was much lower than it is today. So part of that could be that the population has just expanded and reoccupied habitat. The other potential explanation is we know that with climate change that habitat is moving north. And so it's probably some combination of increased numbers of sea lions and the optimal habitat may be changing, may be increasingly going to the north, moving northward. But you know, we would have 20 years ago, I would have never thought about having the ability to tag female sea lions at Año Nuevo. And so we're going to be we're going to put out 12 more tags in the end of October. And I'm quite excited to see how female sea lions are doing because this is new habitat for them. And we've got lots of data from Southern California, from San Nicolas and San Miguel. So it's going to be a really interesting comparison.
Very good. I think we have one last question, and then we could probably wrap it up there. But we have regarding delayed implantation. Do you have any theory about what the biochemical signal is that instructs the embryo to drop down the fallopian tube to implant in the uterus?
Well, yes. Well, actually, we had this is an interesting story. We had a human fertility doctor from University of California in San Francisco, UCSF, who wanted to study that very question. And what basically the egg goes into the uterus and is fertilized, and what it does is it floats around uh, in the uterus and then it implants. We're not sure what the signals are. We we've been studying, we've been doing the hormonal studies to see when actual implantation occurs. We think it's during the molt. We had proposed to do some work on that where we would take some tissue samples from the uterine wall to see what's changing biochemically. Getting the permits to do that became, after a year of trying, I finally quit. And it was, I'll just say the human fertility doctor said these are procedures we do in humans all the time, but we got tremendous pushback in the permitting, and it just became we decided not to pursue it.
Okay. Well, very good. Well, thank you again, Dr. Costa. We're getting a lot of thanks in the chat. Everyone really enjoyed your presentation, and we thank you for your time, and thank you everyone for joining. We will post the recording of this presentation on our website in the coming days. So if you want to see it again or want to share it along, check it out. So thank you again, everyone. Thank you, Dr. Costa.
You're welcome. It's been a pleasure.
All right. Good night, everyone.